My guest today is Professor Karla Kaun, who is Associate Professor of Neuroscience at Brown University. Her research interests include synaptic networks of reward and addiction, cognitive decline, and effects of aging on substance abuse. Welcome, Karla.
Thank you. It's a pleasure to be here.
Yeah, thanks for doing this. So I have a number of your papers, and I want to start with alcohol and drugs rewire your brain by changing how your genes work. Research is investigating how to counteract addiction's effects. You say here many people are wired to seek and respond to rewards. Your brain interprets food as rewarding when you're hungry and water as rewarding when you're thirsty. But addictive substances like alcohol and drugs of abuse, can overwhelm the natural reward pathways in your brain, resulting in intolerable cravings and reduced impulse control. So before I get into the details of this, Carla, so I want to sort of, as I mentioned, I don't really have much of a background in neuroscience or neurology, um, so I want to sort of understand the brain a little bit. And we're going to use the fly model here, and you say the fly brain is somewhat similar to the human brain. I find this really fascinating. So the fly has about 100,000 neurons approximately,
hmm
and so I was just looking at these neurons, and I was thinking it seems like evolution gone bad in some sense because if I were to design a computer, I will have a central processing unit and have a bunch of, electrical circuits running into it. But this is some sort of a distributed processing system, right? So all these neurons sort of talk to each other and try to compute. And so what is the scale that we are talking about? This 100,000 neurons, how big are they, each of these neurons?
So the neurons themselves are very small. Like the cell body itself is maybe a couple microns for most of the neurons, and the brain is organized in a little bit of a different way. It's a small brain problem. So all insects, for example, in our brains we have. So the way a neuron is made is you have a cell body, and then you have things projecting from the cell body that are called dendrites, and they receive signals. And then you have other projections from the cell body that give the signals, and those are called axons. And so
Yeah.
in on all in things with very, very tiny brains like invertebrates, the cells are structured slightly differently. You have a cell body, and then you have the dendrites and axons are sort of projecting in there's like in inwards towards the middle of the brain. So all the connections are made in the inside of the brain, as opposed to like the dendrites being here and the axons being here, as they often are in mammalian brains. That being said, the neurons work a lot like our neurons, and they're exactly like you said. Basically, like it's all about connectivity and how
Hmm.
each of these tiny little components or cells is attached to other types of cells, and how they give signals and then how they receive signals. And so clearly, there's leagues more complexity in our brains compared to fly brains. But with only 100,000 neurons, flies can do crazy things, right? They
Hmm.
learn, they remember things, they can avoid predators, they can fly. We can't fly. They can do
Yeah.
a tons of different types of behaviors, and their behaviors are surprisingly complex. The more we understand about them, the more I think scientists in general are just blown away by how complex these types of creatures are. They have very rich social behaviors.
Mhm.
And they have really complicated foraging behaviors, for example. And so, with only 100,000 neurons connected in the way that they can mask a lot of different types of behaviors.
So understand the hardware a little bit more. So there is a cell body,
Yes.
and there are, I guess it's called dendrites. So that's how the information is coming into the cell body.
hmm
And then axons sort of take information away from the cell body to the next one.
Yep.
And what puzzles me is that, the transmission at the end of that axon is by neurotransmitters. So this is a sort of an electrochemical system. Do we know if there is any magnetism involved here in any way, or just electrochemical?
Oh, that's really interesting you bring up. There's actually some controversy in the literature about whether there are neurons that are sensitive to magnetic fields. And so it's not my area, so I couldn't really comment on that. But I think the more we understand, the more we don't understand, if that makes any sense. So the more things we learn, the more fascinating things get. And in truth, we don't know all that much exactly about how the electrical side of neurons work, because there's, all the neurons are attached to each other with these little electrical gaps. And we don't really know that much about how those types of things work. A lot of us in the neurobiology realm, they focus more on the chemical side of the synapse. And we know we need the electrical connectivity to result in the chemicals moving from one to the other. And that's sort of like how the neurons signal to each other. But there's just so much that we don't understand.
So that's sort of taken for granted. And then you have neurotransmitters at the end of it that sort of transmits information to the next one. So I was thinking from a design perspective, this appears to be very, very inefficient.
Well,
But
it is inefficient, but it's actually really smart, because it turns out that most neurons don't have just one neurotransmitter. They have multiple neurotransmitters. And so that's brilliant, because it can create a flexible signal. Like with one type of activation, you can release maybe just one type of neurotransmitter, and with another type, you can release the other type, or you can have one that releases both types, but they go to different places. So I think it's a really brilliant way to get, a simple thing to do multiple tasks.
So the neurotransmitters that are released, is that a function of the electric potential that's coming to that? How does it work?
It is and it isn't. So what happens is you get these ions moving into the cell, like calcium, for example. And when you get a lot of calcium moving from the outside of the cell to the inside of the cell, that will change the electric current. And when there's enough calcium amassed, basically, you get chemical reactions and enzymatic reactions happening that result in vesicles sort of moving to the tip, and then the transmitters getting released from the tips of the axons onto the next cell.
Yeah, so just talk about this. So alcohol and drugs, you say rewire your brain by changing how your genes work. So genes is sort of a component of your DNA, and so the DNA is sort of an assembly of genes. Is that the way to think about it?
Yeah. So I'm going to go all the way back to the very start of every organism. You start out as one cell. And in that cell, you get DNA from mom and you get DNA from dad. And then those two copies come together to make the genetic component of that cell. And then that cell duplicates so many times to eventually make a whole organism. And so each one of those cells has that same copy of DNA. And genes are just sort of subsections, or like little tiny red components within DNA. So it's like DNA is the storybook and genes are like the paragraphs.
So is the sequence of genes on the DNA important, or are they sort of independently working?
That's a very complicated answer, actually, because there's actually genes nested inside of other genes. So the sequence itself is important. And then you have at the start of the gene, you have a section where this enzyme can bind, and that results in replication of that gene so that it can get made into something called mRNA, which is the next step molecule. So it's how the message basically gets read out, and that mRNA gets made into protein. So some genes have multiple start sites, and sometimes there's like a little baby gene inserted within a larger gene, and DNA can be read both ways. It can be read this way, and then it can also be read this way. And so sometimes there's a gene going backwards within a gene, for example. And so that's a very efficient way to create a lot of different genes within a small subsection of DNA, basically.
Yeah, so I mean, you don't have a lot of space, so you have to very efficiently organize inside the cell.
Yes.
So I don't know much about this, Karla. So mRNA. So I have heard a lot about this mRNA vaccine as part of the COVID-19.
Yeah.
So what is the connection there?
So our bodies function because of little proteins, which are basically like molecules that do all the work in our cells. And the proteins are encoded by the genes, but there's this step in between. So the gene gets expressed as mRNA, which is like a single strand molecule. And then that mRNA gets taken out of the nucleus of the cell, which is where all the DNA is. And then it gets transported to a ribosome, and then it gets made into a protein. And so there's 3 tiny little base pairs are read into one amino acid, and each protein is a whole bunch of amino acids put together into one molecule. And so mRNA is sort of the transitional stage between gene and protein.
Okay. So, so how do alcohol and drugs rewire the brain? So what do we know?
There's many different ways that they can do it, but I'd like to talk a little bit about how I think they affect how the genes are expressed. Okay, so you're born with a certain set of genes, basically, and those don't change, right? But what do change is how they get turned on and off. And the way that they get turned on and off can be very dependent on each different cell type that you have. And there's many different cell types. There's hair cell types and skin cell types and brain cell types. And so each of those different cell types, they all have the same genes, but the genes are expressed or turned on or off at different times. And that results in the cell looking so different during development. And then all your genes are also very active as adults too, because your proteins get constantly turned over, right? And so each cell is always making protein, and then some of that protein is then like getting degraded and new protein is getting made. In order for new protein to get made, your genes have to turn on and off. And so what alcohol and drugs of abuse, pretty much anyone that anybody's ever looked at this point, what they do is they can change how genes are getting turned on or off, and then how much of that gene is made, for example. And so this is sort of a whole new understanding that we're starting to have of the brain. We didn't realize, I think, for a really long time, how dynamic gene expression was. We thought, okay, once your neuron is this type of neuron, this is just kind of all the genes kind of stay like this. And it turns out that that's not what happens at all. In the context of memory formation, for example, you need structural changes to happen in a cell. You need more synapses to get made or less synapses to retract. In order for those structural changes to happen, you often need different kinds of molecules getting made. In order for those proteins and molecules to get made, the genes have to turn on or off. So it's an incredibly dynamic process. And basically, alcohol and drugs are sort of tapping into that, and they're cranking it up like a volume knob. So they turn it up or they turn it down, depending on where in the brain we're looking at.
Yes, it's really fascinating, Karla. So I was thinking, you mentioned flexibility. So it seems like genes can go in a lot of different directions. It's not a computing unit the way that we think about in computer science. Given different inputs, they can have different behaviors. They can produce different things. So it's a bit like a factory that could produce cars, airplanes, depending on what the inputs are. Is that the way to think about it?
That's a really interesting way to think about it, actually. I never thought about it this way. So yes and no, because, you're going to encode all of the same essential components, like, all of the same components need to be there in order for the cell to work. But certain other things could be made. For example, say you're a factory that makes all kinds of vehicles, including like cars and trucks and hybrids and electric vehicles. Yeah.
Yes.
Okay.
Yes.
So you make all of these different types of vehicles, and you'll have certain subsets of genes that will make a truck and certain subsets of genes that'll make a car. But then maybe you have, in order to make truck number, like a big pickup truck versus like a tiny little electric truck, for example, you would have the same genes, but there's different versions of those genes that can get expressed to make one type of truck versus another type of truck. So that allows a lot of flexibility.
Sorry to interrupt. So different versions of the gene. And so that's a hardware change, right? So let's say I'm born with gene, X, and then over time, and we talk about alcohol and drugs here, so we get the details of this. Over time, that X is turned into X prime. So it's sort of a different factory now. It used to produce gas vehicles, now it's
Mhm.
producing electric vehicles, or something along those
Yes,
lines.
yes and no. The hardware, all the DNA is the same. It's that
Okay.
transitional stage. So one gene can make many different versions of the proteins with slightly different tweaks. Green car, red car, yellow car, blue car. And those are based on, so you have many different mRNAs that can come out. And it's because genes are made of multiple different points or different
Yeah.
chunks. And these chunks are like exons. Yeah. And
Yeah.
so a gene for one molecule might have 8 potential exons, but sometimes one of those exons gets left out. And so you only have 7 exons. And so that'll result in a green truck. But maybe, all 8 exons get made and that's a blue truck, for example. And so it's sort of like there's multiple layers of sort of complexity built in, which allows this kind of flexibility.
Yeah. So, I mean, it's very intuitive that people are wired to seek and respond to rewards. And I mean, we all know this. People get a lot of pleasure from alcohol and possibly drugs. And so those things are rewarded. And if I understand this correctly, Carla, the expression of the genes over time changes. It's sort of conditioning the gene to do something different, right, over time. Is that what's happening?
Yeah, and it is very dependent on, which type of neuron you're looking at and which brain area you're looking at. And so what we know now is, so I have to tell you a little bit more cell biology, bear with
Yeah,
me.
yeah,
So we
absolutely.
have
Yeah.
all of this DNA in cells, but as you mentioned, it's really hard to fit this amount of stuff inside this tiny little nucleus. And so the way
Yes.
that we do this is that the DNA gets wound around these proteins called histones, and so it gets wound up and then compacted. And
Like a
so
bit like a spindle or something
kind
like
of like,
that.
yeah, that's actually
Yeah.
a really good way of putting it. So you have like DNA wound around each, and then the spindles get all compacted together. And so it's really,
Okay.
and when it gets compacted like this, the genes can't really turn on because
Hmm.
there's no space to allow those proteins to bind to them, which help turn them on.
So no
And
signals. They're sort of shielded some
basically,
way.
yes. So,
Yeah.
so what drugs and alcohol can do, we know now from animal models. We assume that it also happens like this in humans, but we can't look at that. um, is it allows different sections of the DNA to unwind or to wind tighter. And so what it can do is it can control basically, which groups of genes get turned on and off. So that's one layer of regulation. Another layer of regulation is it can change. You know how I was telling you that there's different exons that make blue cars or green cars? It can change whether the car is blue or the car is green by changing the number of exons, for example, that stay in the mRNA. And it can also, once the mRNA gets made into a protein, those proteins have lots of tags on them that are basically like instructions for where that protein needs to go and what it needs to do. It can
Yeah.
change all the tags on the proteins. And so
Okay.
there's so many ways that drugs and alcohol can do this. We don't know necessarily exactly how all of that happens, but we know that it does happen. And there's scientists all over the world that are sort of actively investigating how this happens. Is there a common mechanism? Is it like this in all animals? Is it dependent on how active the cells are, for example? And because neuronal activity is a very important marker for how much reward we'll feel and how what kind of memories we make and these kinds of things.
So I know there's some correlation between alcohol consumption and cancer. And if you say cancer is sort of a mistake in transcription or translation, or I don't know what the right term is. So when you change the factory, do you have a higher chance of errors in production?
So what happens is you're not changing the factor, you're changing the workers.
Yeah,
So
changing the
so
workers.
so
Yeah.
what happens is alcohol can change the cell signaling pathways inside of a cell. So in the outsides of the cells, you have all of these sort of receptors and molecules sort of like hanging out on the outsides of the cell bodies, and some of these have tiny little tags on the inside that when they're broken off, they go to the nucleus and they turn genes on and off. And so one of these cell signaling pathways that I study in my lab actually is called the Notch signaling pathway. And we know that alcohol can result in more of the inside part of Notch getting broken off and going to the nucleus and changing gene transcription. It turns out that Notch is really important in cancer, for example. And so it could be, we don't know, but it could be that one of the ways that alcohol can affect cancer, for example, is by messing with signaling pathways like Notch. And there's not just Notch, there's so many signaling pathways. They've traditionally been studied in the context of development in cancer, and they haven't traditionally been studied in the context of how adult brains function. But it turns out all of these things are still in the brain. They haven't gone away. We just haven't really studied them before because they're really hard to study and complex, and we didn't know that they were this dynamic or active in adult brains until fairly recently.
Yeah, so talk a little bit about the fly. You have another paper here, Drosophila reward circuits. The ability to sense and respond to a rewarding stimulus is a key advantage for animals in their natural environment. The circuits that mediate this response are complex, and it has been difficult to identify the fundamental principles of reward structure and function. So we know these things exist. As humans, we experience them all the time. But yeah, as you say here, it's a very complex phenomenon, isn't it? I mean, some electrical signals have to travel from X to Y, and some neurotransmitters have to start going in one direction. So what is our current understanding of how the reward mechanism works?
Oh, that's a big question. Many people around the world are trying to answer it. We know that we have centers of the brain that are more sensitive to rewards. And like all types of sort of behaviors that happen in the brain, it probably requires all brain regions, but there are sort of control centers. Our brains are sort of structured in all of these like small like components and structures and stuff, and fly brains are too. All brains are. And it turns out that a lot of the structures that we think are important and activated in reward have a neurotransmitter dopamine and a whole bunch of neuropeptides that are a little bit longer acting than dopamine. And we know that when we experience reward, even in a natural context, it increases the amount of dopamine that gets released.
And dopamine, dopamine is a neurotransmitter, or?
Dopamine is a neurotransmitter. Yes, and it's one that it's actually really important in general brain function, not just in reward. So it's not like the reward molecule. We actually don't know what the reward molecule is. It's probably a combination of very complex factors. But we do know that dopamine is released in reward, and dopamine is very important in memory as well. So reward and memory are very, very closely intertwined because our brain sort of evolved to remember where the good stuff was.
Yeah.
So every time we're experiencing reward, we're probably also making memories about that experience as well. Maybe the memories are sort of passive, like the sensory experiences, the feeling of our clothes on our skin, or the smell in the air, or the people that we're with. And maybe they're also active because we're actively working to get more reward. So it's almost impossible to dissociate reward and memory mechanisms in our brain. But we do know that dopamine gets released, and then that talks to the cells that are sort of the next in line. And then those cells can help initiate an action. And what happens over time when you experience a lot more reward or more signal is that the structure in those cells starts to change a little bit. Maybe the connections get tighter, or maybe the molecules change to result in faster connectivity, for example. And so that's what plasticity is, basically. It just means changes that are happening to the components in cells or to the structure of cells that result in things firing faster or in a different way to elicit one particular behavior over another. Now, you asked about flies. Why on earth would we study this in the context of flies, right? And the reason why is because they only have 100,000 neurons. And we have an electron microscopy level of resolution over the entire fly brain. So what this allows us basically is a really, really, really specific map of exactly how every neuron is connected to every other neuron. And it's important to have that kind of map because it turns out that it's really important to know how neurons are connected in order to understand how functional behavior is happening. Things are
So that's
way more complex than we thought they were. One neuron can activate hundreds of other neurons. And so
yeah
how could we possibly understand that in our brains, which are, exponentially more complex without understanding
yeah.
it first in a
And we have something like 87 billion neurons.
Yes, exactly,
Yeah.
with so many different connections.
So I want to go back to dopamine. More fundamentally, how are memories stored? Is the memory in the neuron or the memory elsewhere?
That is an excellent question. I you ask a different memory scientist this question, you'll get a different answer. So I'm going to cop out and say that I think that there's probably multiple levels of memory that's being stored. We do know there's something called an engram that happens in a brain when a memory is made. And the engram is thought to be sort of the physiological correlate to where that memory is stored. And it's a combination of a number of cells. It's not just one cell. It's a bunch of cells that get reactivated whenever the memory gets reactivated.
So it's a
Okay.
configuration. It's a configuration of cells. Is that
It's
how
a
the
it's
memory?
a configuration of cells, and there's probably something really interesting about those cells that results in just those cells getting activated for just that memory. It has to do with how those cells are connected to all of the things that were sort of active when the memory was getting made. It has to do with how active that cell is naturally. So some cells get activated really easy and some cells
Hmm.
don't. So it has to do with that. And it probably has to do with sort of the molecular underpinnings of what's going on in that cell. Because we do know that in order for structural changes to happen, molecules have to change. And so there are transcriptional mechanisms, meaning gene expression changes, that are happening in those cells that are resulting in those cells being the memory cells or the engram cells. And so I think there's multiple layers. There's like networks of things that are webbed, that are associated with all of the things you're experiencing when that memory is getting made. There's molecular networks happening that change inside of those cells, but it is some sort of combination of cells that is associated with one particular type of memory.
Yeah, I mean, just like a hard drive, there's like sort of magnetic stuff going on there. It's a bit like that, right? So you have a configuration that is in the brain and that sort of points to a memory. So going back to the dopamine, is it facilitating memory formation, or so? What exactly is dopamine doing in that process? It's a neurotransmitter, right? Yeah.
Yeah, so dopamine is doing a number of different things. One is that when the reward is experienced, dopamine neurons are initially active, and they're part of, some of those cells are part of the engram, and some of them are activating cells that are part of the engram. And so what's interesting about dopamine in the context of reward and addiction, is that when you get more of the reward and you're going to get used to the reward, the dopamine doesn't fire as much. And what fires isn't dopamine, but all of those cells that dopamine was signaling to. And so the memory gets transferred. So it's not just a hardware thing, because it's called systems memory consolidation, because it changes in the brain as your memory gets made and as it gets re-experienced. So it's almost like the engrams are dynamic in some ways, and in other ways they aren't. In truth, we don't actually know from beginning to end, at least not in mammalian brains. In fly brains, we're basically picking apart the circuits one neuron at a time to be able to figure out exactly what's going on when this happens.
Yeah, I mean, I'm thinking, Karla, there should be some therapeutic stuff here. Often we just turn the computer on, computer off, and then turn it back on. And
If
that
only
that
that would
seems to
work in humans.
that seems to solve a lot of the problems. But in this case, these are like permanent memories, so we cannot really turn it off. But we could reprogram it back, could we?
So that's the idea, right? Like if we could figure out exactly which cells were getting active when, we could reactivate those cells and initiate the memory, or we could silence those cells if it's a memory that we don't want, like a really bad trauma.
Yeah.
And so we can do this optogenetically in flies and in mice. We can use genetics to turn cell activity of cells on and off, and we can create false memories. by doing this, or we can reactivate a memory, or we can silence a memory. We wouldn't want to do that in humans necessarily. That's an like science fiction level of control. That's non-ethical. But maybe someone has post-traumatic stress disorder and they want to be able to reduce some components of their memories. Is there a pharmacological way that we could do this?
Yeah.
And I think that in my lifetime, we might get to a stage where this is possible, because we're starting to understand more and more. The problem, of course, is that the more we understand, the more that we don't know because of the complexity of molecules in these cells. Like which pathway do you target? How do you silence just a small subset of cells when sometimes the memories change from one structure to another structure? So there's still a lot we don't know, but I think now that we understand more about sort of dynamic gene regulation in the brain, we'll be able to start addressing these types of questions.
Yeah, PTSD is very interesting. So I remember, Karla, this was a few years ago. I was reading something like when you have a really bad situation, somebody dying or something like that, you have less of a PTSD issue if you drink a lot post the event. Is there any connection there?
So I'm not familiar with this study, so I don't know about this, but alcohol certainly taps into memory circuits in lots of different ways.
Yeah.
So one of the ways that it does, and the way that we often study it in my lab, is when you're drinking or when you're being intoxicated by any pharmacological substances, you're making memories of that experience. Maybe it's like the feeling of the glass in your hand or the bouquet of the particular wine, but it's like a sensory experience that gets associated with the feeling of being intoxicated, and that results in a very strong memory, right? And so when you smell that wine again, you might get a craving for drinking. So that's one type of way that alcohol and drugs can tap into a memory. Another, of course, is that they affect many different areas of the brain, and they certainly affect areas that make memories while you're being intoxicated, and they affect activity of those levels. So while you're intoxicated, you're not going to remember events that happened during that time in the same way, because your internal state is different. The activation parameters in your memory structures is different. How you're feeling emotion is different. Emotion is also very important for memory. So it messes with memory that way. And then the sort of more severe kind is you get blackouts, right? Because it messes with the memory so much that your neurons basically stop functioning, and you basically go into an unconscious state. And so that part of it, we actually don't understand very much about that at all.
And blackouts. So the human is still functioning, like the person can walk around and do whatever, and then the next morning the human doesn't remember what happened the evening before, right? That is the definition of blackout.
Yeah, well, I mean, there's the kind where you just pass out and you're sedate, blackout, and then there's the kind of where you had a great night out and then you wake up and you don't remember any of it. Yeah, in truth, we don't understand very much about how that happens, but we do know that alcohol in particular affects pretty much any area of the brain that anybody's ever looked at. It just affects different cell types in slightly different ways. And so until we really understand how memory is formed, we probably won't be able to understand the extent through which alcohol affects formation of different types of memories.
Yeah, I mean, you have done a lot of interesting studies around flies. So I want to touch on some of the studies, Karla. So before we get there, I just want to ask you, I was just thinking, you find the fly brain very similar, if I understand this correctly, to human brain. But in pharmaceutical industry, we don't use flies for any tests. We use dogs and chimps and things. Do you think pharmaceutical is it just the brain that's similar, not the body? Is that the right thing?
Well, I think that there's some things that are similar and there's some things that are different. What I think we've discovered in the last 20 years is that the structure of the fly brain is organized in a way that is phenomenally similar to mammalian brain. Like we really thought, when I was in graduate school and before that, we really thought fly brains were very different. And there wasn't a point to really studying circuitry in flies. And the field has basically gone 180 on that, because the more we understand about that, we're like, oh, there's this beautiful reward structure that looks a lot like ours. There's this beautiful memory structure that looks a lot like ours. So their brains aren't as complex as ours, but there are, like, the way that the neurons are organized into a structure is very similar. And so the structure of the fly brain that I look at, for example, it has a similar structure as many different structures in our brain. It's just
It's
sort of
what you
like
call the
basic components are there.
mushroom body or
The
whatever?
mushroom body, yeah.
Oh, okay.
Yeah. And so it's like a structure is completely innervated by dopamine, and it encodes sensory experiences, and then it takes those signals and it relays them to other areas of the brain. So the way that area is organized is not necessarily unlike the VTA to striatum circuit or the amygdala circuit, or even some circuits within the hippocampus. There's a lot of similarities between the different types of circuits. So it's not exactly like our brains, because our brains are just bigger. There's phenomenally more neurons, there's more connections, but they have components that are similar, which means that you can use the fly sort of as a simpler way to study just how this structure might work, or just how this particular process might happen. So that being said, I think flies are actually a really fantastic in vivo model. And by in vivo, I mean in whole organism.
Yeah.
And I think that one of the things that pharmaceutical companies could do better is to use in vivo models, because cells in a dish don't work like cells in a brain.
Never work.
And you can actually screen through drugs quite quickly in things like worms and flies that have a functioning brain inside of a whole body, because the brain body axis is really, really important for understanding how drugs work. So flies' bodies, I mean, they have all of the same internal structures that we do. Their circulatory system is a bit different. They have an open circulatory system instead of a closed one, so they don't have like veins and arteries the way that we do. And their respiratory system works a little bit differently, but the same components, like how a cell works, is the same. And their feeding and their insulin signaling pathways is remarkably like ours as well. So I think there's so much potential that's untapped. And part of the reason, I think that some companies have used flies before, but then they're like, oh, these are too complex. And then they scrap those projects. But I would argue that it's actually one of the reasons why a lot of drugs don't work, is because they're not using a complex enough system. They're using cells in a dish that just act very different than cells in a brain. And if you screened in a fly, maybe more of the drugs that you found would work in other organisms as well.
Yeah, that makes a lot of sense. I mean, we have solved all of the physical problems, but we haven't solved the CNS problems. And it seems like for CNS, fly could be a very, very interesting early model to look at things, right?
Absolutely. And a fantastic model for disease, because, we can take a human mutation, for example, ApoE4 in humans is associated with Alzheimer's. We can take that human gene, plunk it in a fly, in the fly brain, and then screen drugs and in like a working brain that has the same sort of, ApoE induced type problems. And it would just happen so much faster than if you were to do it in mice.
Do we see in flies, I mean, I'm going on a tangent here, Karla, so stop me if we don't want to go there. But do we see things like Alzheimer's and Parkinson's and things like that in the fly brain?
It's traditionally been a little bit difficult to study that. So the flies are actually a fantastic model for aging, because they age pretty quickly.
Yeah.
But and so like a 60-year-old fly, so flies in nature will last maybe 2 weeks. They basically live enough to reproduce in a few days past that.
Just 2 weeks?
Yeah.
Just 2 weeks?
They have a lot of natural predators, and life is hard out in the real world.
Yes.
In the lab, they have very privileged lives, if you will. And in longevity studies, we give fresh food to the flies all of the time. So they live up to 60, even up to 90 days. There's a mutant called Methuselah that lasts like extraordinarily long. And so this is nice because you have a very sort of fast model of aging. But people haven't really seen sort of like the natural plaques or fibrillary tangles in even old flies that we see in super aged people yet, but you can cause them by taking the mutations that are associated with those diseases in humans and putting them in flies. And you can cause the same sort of things. So what you're going to do is you're going to take like an aged fly and combine it with a genetic model to be able to model these types of things. So it doesn't necessarily naturally happen, and we don't know why. It could be that you just need more than 90 days to get these types of things, right? It could just be time itself. We don't know.
And I know that you do some work in aging. I haven't picked up any papers, Karla, but so is aging related to the brain or the body?
Wow. So the aging stuff that I do is it's a very, very new part of my lab. And part of the reason that I started getting involved in is there's amazing aging researchers here at Brown. And so basically just by hanging around these people, I got really interested in questions that involve these types of things. And I think the way that people sort of studying it is really, we can't really separate out the brain versus the body without looking at just sort of the general cell biology. And an old cell looks and acts really differently than a young cell. And an old neuron is different than an old muscle cell, which is different than an old like lung cell, which is different than an old skin cell. And so I think it's probably everything. And it has to do with sort of probably common signaling pathways within all of these cells and how they might act slightly differently in different contexts.
Yeah, I mean, the brain part of this is very interesting. So if you live for a while, your neurons have been working, they get all sorts of information, they put out some information, they collaborate, they do stuff. And over time, in any physical system, you have issues. You don't do the transmission as fast, or whatever the case may be, right? So that's sort of part of aging, right? So if that is true, then we could have a therapeutic intervention in the brain to sort of put them back in good shape, wouldn't we?
Make a young brain. Isn't that the dream? I get the distinct feeling that this is one of the major goals of sort of the aging field. I don't know if I necessarily agree with all of that. I'm quite happily aging. Life gets older as you get old. Life gets better as you get older, as far as I'm concerned. But I think it's certainly possible, and certainly in my world where we're dealing with flies, and we can manipulate genes so easily. We make old flies young by just messing with their gene expression in different ways. So,
can you make a fly more intelligent by manipulating the neurons?
So this is a complicated question because it really depends on what you mean by intelligence.
Yes.
Most of the fly memory field, that's sort of like, most of what people consider intelligence in flies is the ability to learn faster or more.
Yeah.
And so there's basically like one main type of memory that people study in flies, and that's the ability to associate a sensory cue like a smell or a visual cue with something that's bad like an electric shock, or good like sugar. Okay, so flies will form this association. If they have a smell with a shock, they'll avoid that smell. If they have a smell with sugar, they'll go towards that smell. And that's it's called a Pavlovian memory, and it's a very sort of simple associative type of memory. You can make flies remember things for really, really long periods of time. And you can make flies learn faster. And you can make flies learn more things at once. So if you want to define all of that as intelligence, then yes, you can make more intelligent flies. But I think that intelligence is much, much more complex than that, personally.
Yeah, it's very complex. Yeah.
So, but I think you can definitely mess with memory.
Yeah. But can they learn time? So more tactically. So suppose I have a sugar fountain that turns off at 2 p.m., comes back at 4 p.m. Will they be able to learn that?
Yes. Yes, people have shown that. Flies, actually, a lot of what we understand about circadian rhythms or how our bodies respond to time comes from flies. The Nobel Prize in 2017 was awarded for that work. And so we know a lot about that. And they learn different things, and they sense different things at different times, and they actually have a sense of time. And so the answer to that is definitively yes.
Yeah. So we talked a little bit about this, so but I want to get into sort of the metrics here. So you say just a few drinks can change how memories are formed. We
Mhm.
talked a bit about this. So what do you mean by just a few drinks? I mean, this metric has sort of moved over time. What people considered a few drinks maybe 50 years ago is very different from what young people consider few drinks today. So what exactly is a few drinks today?
The way that I'd sort of define that is that I think uh, it turns out that very low doses of alcohol, or just one drink, for example, can create way more changes than we thought it was.
Hmm.
And so I think, if you looked in the literature even 20 years ago, you would think that only really high doses of alcohol could cause lots of changes. But it turns out that a lot of that work for low doses was suppressed because some of the funding came from the alcohol industry. Uh, so there's some like complicated social factors. And so the stuff that's coming out now that is, more government funded shows that even low doses can have pretty extreme effects on your physiology and your brain.
Hmm.
And what we've shown in the lab with flies, for example, is that the equivalent of 3 drinks, where each drink was consumed within, say, 10 minutes and then spaced by an hour, so 3 drinks over an evening,
Yeah.
is enough to change activity of a cell signaling pathway that's really important for dopamine signaling up to a day later. And 2 drinks wasn't enough. So my recommendation is, if flies are anything like humans, and
Yeah.
they have the same signaling pathway, but we don't know if it works like this in humans, I would say, 2-0 is the limit before
Yeah.
you start affecting the memory pathways.
It's surprising because alcohol has been sort of naturally occurring compound that all animals, and including mammals, have had exposure to for a long time.
Mhm.
So it's quite surprising that there's such negative effects um, on humans. Um, I'm just
Well,
saying, yeah.
I think that's an interesting way to think about it. But I come from sort of, an ecology and a neuroethology sort of background, and so I'm gonna tell you sort of my perspective on things. Every plant, and alcohol's a little different. I'll talk about that in a sec. But every plant that produces a drug, like opium comes from poppies, and cocaine comes from coca plants, and nicotine comes from tobacco.
Yeah.
They were each produced. Each of these substances was produced to get rid of insects,
Yeah.
because
Yeah.
they're pesticides by causing neurotoxic effects on the insects. Basically, they like paralyze their nervous system.
Yeah.
um and and each of these, that they cause, pretty profound pharmacological effects on the
Hmm.
brain. And so I would say, just because it happens in nature doesn't mean that it isn't bad for you, right? And it could have evolved for a reason completely different. Like, it certainly, tobacco evolved to affect insects that didn't evolve to affect humans at all. We just
Hmm.
use it as sort of like a byproduct, because as humans, we liked the feeling of nicotine as a stimulant. Alcohol is very interesting because it's not actually produced by a plant the way that these are to get rid of insects. It's a byproduct of fermentation,
Yeah.
right? And so insects and mammals figured that out a long time ago, and they liked it also because it gives calories. The other types of pharmacological substances don't necessarily give calories, so you get a double whammy. One is calories, and our brains like calories, and the second is the pharmacological properties of it. And it turns out humans like that too. And so
Hmm.
I think that, we're basically tapping into our natural reward circuits. And a lot of our natural reward circuits were evolved to like food, basically, because food
Hmm.
is what we need to survive as a developing and as an adult animal. And so I think that probably there's a long history of humans using alcohol, but I don't
Yeah.
think it's not necessarily surprising that we use something that is naturally found in our environment for our own pleasurable reasons.
And I was reading, Carla, I think maybe your papers or others that there's sort of a genetic, so there is a gene that gives you a higher pleasure for alcohol consumption.
Mm.
And I grew up in South India, in a small state in South India, and this is the per capita alcohol consumption is one of the highest in the world there.
I didn't know that.
Yeah, so it's sort of interesting. So it goes back to the reward system question.
Mhm.
If your brain is rewarding you, even for small amounts of alcohol, you'll keep going to higher and higher levels. I mean, I don't know this, but I wondered if this is in fact true from a genetic basis, then evolutionarily you would have done something to counteract it, right? I mean, maybe some sort of physiological change, or I'm just thinking. I wonder, yeah.
So, you have to remember too that evolution only happens by reproduction. And so if it's not affecting reproduction, it probably won't change over evolutionary time.
Right.
That being said, there are natural variants for lots of different genes in the world that affect human preference for alcohol, or at least the way that humans metabolize alcohol. Like
Yeah.
the flushing gene,
Yeah.
so there's variants for that, and there's variants for another enzyme that's in a very
So they
similar
cannot drink.
pathway.
They cannot drink much. It's sort of a defense mechanism,
Yeah,
so to
and
speak.
for
Right?
some
Yeah.
people it makes them more sick, and for and other people it allows you to be more tolerant. And the one thing that's correlated with an alcohol use disorder is how much alcohol you can consume. You need a lot of alcohol to develop an alcohol use disorder, and if you have a high tolerance, you drink more, which means there's more opportunity for alcohol to affect your brain circuits.
Yeah.
Right? And so a higher tolerance is thought to be associated with increased
And
preference, or at least increased consumption, if not preference,
so
and metabolism can contribute to that.
is tolerance sort of a physical thing? Meaning, if I have no tolerance and I consume more than what I can tolerate, I'll throw up. Is it a physical reaction to what I
It's
could
both.
consume? Yeah.
Yeah. So there's tolerance that happens within your circuits, and then there's
Okay.
tolerance that happens specifically even within your reward circuits, and there's also physiological tolerance. And we don't understand exactly all the mechanisms. Actually, flies have been a fantastic model for studying tolerance, because it's
Yeah.
basically like comparing the early versus the later use, and you can do all kinds of cool screens and discover mutants. And I even go into schools and do these experiments with kids so that they can test different mutants and how they affect these types of things.
Yeah.
Mhm.
Yeah, so I want to finish up with, we're running out of time. So I want to finish up with one of the papers, I think from 2012, sexually rejected flies turn to booze.
Uh-huh.
You say here, offer a male fruit fly a choice between food soaked in alcohol and its non-alcoholic equivalent, and his decision will depend on whether he is mated recently or been rejected by a female. I think this happens in humans too. Um, so,
No
so this
comment.
fundamentally confirms flies are like humans.
I think I think flies are like humans in a lot more ways than we think they are. That study was actually done with a very good friend of mine, Galit Ophir, who we did these when we were postdocs together in Ulrike Heberlein's lab. Ulrike Heberlein has since retired, but she's sort of person that sort of started this whole field. And Galit is continuing to study some of this in her lab, and I continue to study some of it in our lab, and we continue to collaborate. And so it turns out that flies have very complex social behavior, and one of the
Hmm.
most important social behaviors is mating. And mating, Galit showed, is rewarding in flies. And it's not just any aspect of the mating, but copulation itself is
Yeah.
rewarding in flies. And it turns out that copulation activates peptidergic pathways that elicit reward. And alcohol basically taps into these peptidergic pathways
Hmm.
that elicit reward, and then it allows sort of like flies that haven't been mated basically are in a different internal state. Their peptides work differently in their brain, and that
Hmm.
results in them seeking reward in a more extreme way, and so that's why they consume more alcohol.
So is there a dopamine connection here, Carlos? So let's say the fly mated
Mm-hmm.
and its brain is sort of flooded with dopamine. I'm just thinking, and now it doesn't have, it's a different behavior. Is that what's happening? Is it some sort of neurotransmitter release that sort of put the brain into a different state, so to speak?
So we actually know exactly, that's the benefit of using flies, is we know exactly what's happening here.
Okay.
So what happens is when flies are sexually rejected, it changes the levels of neuropeptide F, which in us is called neuropeptide Y. And this puts the flies in sort of a different state. It turns out that neuropeptide Y neurons are activated by dopamine, and dopamine neurons actually have receptors for neuropeptide F receptors in flies. So it's probably some sort of reciprocal interaction. And so we know that indeed courtship does involve dopamine signaling. There's a number of labs looking at that, including the Michael Creighton's lab at Harvard. And so it's a very complex interaction. But what I think is very interesting is dopamine acts in a slightly shorter time scale than these neuropeptides. And so probably it's allowing sort of, an increased flooding of the system by these neuropeptides. And that's sort of changing the basal state of the fly and how it feels. And it's that basal state then that results in the fly wanting to seek reward. And of course, dopamine is very important for wanting to seek reward as well.
Yeah, so your research, from my naive perspective, has a lot of therapeutic possibilities, especially CNS diseases, where we don't have good animal models, the correlations are very, very low to humans. Maybe flies are a better model for CNS diseases early on in animal studies. Would you say, or what do you think?
I think that there's merit to all kinds of studies, but I think flies and invertebrates in general are underutilized. Because you can get to your answers much faster, and their systems are way more similar than I think we ever appreciated before. So the research has a very, I think, good output for being slightly, less time consuming and potentially more inexpensive as well. So I think if people want to use flies for these types of things, there's just so much opportunity. And in fact, myself and other people in the fly community are creating tools to make it easier for anybody who doesn't have experience to just, say like, I want to test this quick behavior. And we're like, okay, great, here, build this. It costs $200 to build it and we'll help you.
And you can do it very, very quickly. Excellent. Yeah. This has been great, Karla. Thanks so much for spending time with me.
Thank you. This was really fun. I really appreciate your questions.
Thank you.